Hello, my name is Annie Slaughter.
And my name is Anthony Dispenza.
And our project is on temperature influencing Acontia production in invasive anemones. Our goals for this project were to understand stress responses in 2 different sea anemone species at varying water temperatures. Some background on this project. It is a continuation of a project done by a graduate from Florida Southern a few years back. In this study, we are exploring the defense mechanism response in these invasive anemones. The 2 species we chose, Diaduma lineata and Exoctasia pallida, are becoming very invasive. D. lineata is spreading from the British coastline across the entire world and is becoming quite the nuisance. And E. pallida is becoming invasive, especially here in Florida off the coastline. So being able to understand what that means invasive-wise is very important. We chose these anemones specifically because they contain Acontia, which is an internal tentacle that anemones will use to defend themselves. Not all anemones have them, but these 2 do, so understanding how their defense mechanism is impacted is very important. This is a new area of study looking into invasive techniques of sea anemones, and not much is known about what they do to ecosystems. And a little bit more on why we chose these anemones. E. pallida is a model for corals, so it's very beneficial to understand how they interact with higher water temperatures so that we can learn how corals may interact with higher water temperatures. And D. lineata, as I stated earlier, is becoming a widespread invasive anemone, so being able to see if it is failing to thrive at higher water temperatures may help us understand its invasive patterns. And for these pictures, we have E. pallida on the left with its Acontia out, and on the right, we have D. lineata under the microscope. Some of the predicted outcomes we believe that the anemones will produce shorter Acontia to conserve energy under stress, but we also thought that we may be seeing longer Acontia produced because they're trying to defend themselves in a stressful environment. For some of the genes we thought we were going to see, we believed we were going to find genes related to survivability being upregulated over just other normal functioning genes. And for our methodology, so we have 2 different methodologies. The first is related to Acontia measurement. So in this diagram, you see that we have anemones from a tank. We took 30 anemones from the E. pallida tank. We put 5 into a Pyrex bowl, 25 into a different Pyrex bowl. We repeated that process with the D. lineata, 5 into their own Pyrex bowl, 25 into their own Pyrex bowl. We then took those bowls and placed them into a water heater. And in the water heater, we had the 10 control on one side and the 50 test on the other. For 1 week, we let them sit and acclimate to 25 degrees Celsius water. At the end of that week, we raised the temperature on the test side up to 30. At the end of the week of acclimation to 30 degrees Celsius, we then took the anemones out of the Pyrex bowls into the 6-well plates, allowed them to acclimate for about 24 hours. And then after those 24 hours, we placed them under a microscope with a little ruler. And using a dissection probe, we very gently would harass them and kind of poke and prod at them until they released their Acontia, where we then measured them at in centimeters. Once we were done measuring them, we took the control anemones, placed them into a Pyrex bowl, and allowed them to recover from being in the water before returning them to their original aquarium. With the test anemones, we then placed them into a Pyrex bowl and placed them back into the water bath at 35 degrees Celsius, where they stayed for 1 more week. At the end of that week, we then placed them back into the 6-well plates, allowed them to acclimate for about 24 hours, and measured them using the same technique again.
So the next step we did, we measured their RNA data. So we first assembled the transcriptome using Trinity stats to attain our N50 values, as well as the total number of genes that were expressed. We then aligned the estimated abundance using Callisto with a pseudo-align. The pseudo-align was used to determine the proper gene expression findings, since the gene expressions profiles for our anemones were not yet created. Once we obtained our alignment with Calypso, we then made a matrix. This allowed us to create, count, and normalize expression values. When the data was normalized, we then used edgeR and dsec to interpret it. We in the R program, sorry, in the XSEDE portal. We then use edgeR to demonstrate the differential gene expression determined by DSeq. Once we obtained our data using differential gene expression, we then obtained a heat map as well as a volcanograph. We then performed a blast using MCVI blast to search our genes from our heat map. The blast search identified genes that were being expressed to determine which genes we would use, which we have listed in our results.
All right, getting to our results. So shown here, we have our length and quantity of Acontia produced, as well as our genes of interest being shown on a logarithmic scale. So diving into our Acontia results, we saw that Ippolita was producing a lot and very long Acontia right away, while Delineata really wasn't, and it kind of went down as water temperature went up. However, after running a Crestwell-Wallace ANOVA test, it was shown that water temperature did not play a role in Acontia production in these anemones. So further studies will have to be done to see if that is actually the case or not. When it comes to these genes of interest,
So what we did was we actually checked. So we did a check on our heat map to determine genes of interest by if they had a greater than 10 value. So when we compared it to our short-term versus long-term as well as our 30 degrees to 35 degrees from our control as well, we picked certain genes that had a higher value. So anything that was greater than 10 is what we looked at.
So after looking at those values that were greater than 10, we found these 47 genes of interest that had been identified, basically. And as you can see, we compared our short-term to long-term data, our long-term to short-term, 25 degrees to 30 degrees, 30 degrees to 25 degrees, control to 25 degrees, control to 30 degrees, control to short-term, and control to long-term. It's kind of hard to see just looking at the graph, but overall, anything from, so our control to long-term and our control to short-term produced the greatest expression over that was greater than 10. And we actually saw the least expression between the 25 degree to 30 degree. Right, so for our conclusion, from the results, we found it can be concluded that with increasing water temperature, anemones will allocate more energy into maintaining cellular and metabolic function over defense mechanisms or other bodily functions, such as producing Acontia. Like I said earlier, our statistics showed that water temperature does not actually play a role in Acontia production, and further studies would have to be done to definitively prove that. Speaking of further projects, for our future project, we would need to analyze Ippolita RNA as well at all these temperatures, and then see if we could connect RNA at 35 degrees Celsius from both species to see if the genes being upregulated change or if they do stay the same.
And for our acknowledgement, we would just like to take this time to thank our research advisor, Dr. Jason McCrander, for helping us with all of our roadblocks we experienced, as well as for all of his help with our bioinformatics troubleshooting. We would also like to thank our advisors, Dr. Gabriel Langford and Dr. Nancy Morvillo, for advising us in the early stages of planning our research project.